So we're going to have some Fluxomics sort of things today. And what I'm going to do is I'm going to go over some data that we've generated over the last couple of years using Fluxomics and how you can derive novel insights into different things by using Fluxomics. And I'll show you a couple examples at least where it really made a big difference to use Fluxomics instead of just using straight metabolomics to understand metabolism. So I need to set up a couple things just to understand it. I'll try and make sure that I'll do the best review of biochemistry I can. So one of the projects that we have going in the lab is to actually understand glucose-stimulated insulin secretion. You would think that this has been worked out, but it hasn't all the way been worked out. But in general, what happens is glucose gets taken up in the cell, and a bunch of stuff happens, and then there's an increase in the ATP and ADP ratio in the cell. And this is thought to close a potassium channel, which causes depolarization of the membrane, opens this gated calcium channel, voltage-gated calcium channel. Calcium comes in, and then membranes fuse, and then you release insulin. Obviously, this is a little bit simplified. There's a number of different aspects to this that seems to play a role in glucose secretion. Insulin secretion. Glucose is actually secreted biphasically. There's a first phase that is likely to be due to this potassium channel event and likely due to ATP generated from the glycolytic pathway. And then there's a second phase, which is probably due to both factors that are generated by glucose and, as I'll show you, probably fatty acids as well, that amplify and cause a longer, more prolonged second phase of insulin secretion. Over the years, with Matt Lorenz and Mahmoud El-Azouni in the lab, we've developed some methods to look at a stable cell line in Swan cells to look at insulin secretion. So here's the first phase of insulin secretion, the second phase. And by doing multiple sampling across time, you can get a pseudo-flux sort of thing, and I'll show you that actually more at the end of the talk. But you can get an idea of how things are changing with time, and most of the time this is pretty straightforward, and the insights that you gain from just looking at straight metabolomics are substantial. But again, it doesn't give you the full story. So we use MS-TOF to do the metabolomics as an accurate mass, and we can usually generate identities of about 100 to 150 different metabolites that are related to core glucose metabolism, some related metabolites. So here's the simple experiment. I'll just show you part of the metabolites that were generated. And so what Matt did in this study is he looked at the fold change in these different metabolites, either with a time course by putting in 20 millimolar glucose or a dose response of glucose at 25 minutes. And if you look at the dose response, you can see that the increase in different glycolytic intermediates goes up in a dose response manner, up to 32-fold increase in the amount of the metabolite. And now this is just without any labels and things like that. TCA cycle metabolites go up in the same way. Interestingly, aspartate, which I'll come back to a couple of times during this talk, goes down in a dose-dependent manner. And long-chain acyl-CoAs actually go down as well. And if you look at the time course, you can see that a lot of these events happen very quickly. The TCA cycle is delayed just a little bit. And in part of the TCA cycle, this probably reflects, as I'll show you a little bit more, is that while the end products, malate, fumarate, go up pretty quickly, some of these intermediates that are more early in the quote TCA cycle go down or go up more slowly. Now, one way to interpret this is that these are coming in and being consumed very quickly, and then you're building up these metabolites at the end. So this is a very fast, and this is very slow. One of the things that most people don't really realize is that the TCA cycle is really not a cycle. It really is two sets of enzymes that act pretty much independently. There is crosstalk between the right side of the TCA cycle and the left side of the TCA cycle. But the right side of the TCA cycle and left side of the TCA cycle probably evolved separately, and that they they subserve different functions in a way. There's a lot of intermediates that are generated from the TCA cycle. For instance, as a lot of you know, citrate or through isocitrate exits the mitochondria at a very high rate and under a lot of different conditions, and that is used for de novo lipogenesis. And the left side of the TCA cycle is used a lot for de novo generation of different kinds of amino acids. And again, while there is interconnection, and this is in a way a cycle, it's not as complete. It's not as complete in sort of a repetitious cycle as one would imagine. Okay, now one way to interpret again this rise in these metabolites, fumarate, malate, and although the fumarate and malate here looks pretty good, like they're coincident, it turns out that we found after the paper was published that the fumarate actually is breaking down into malate in the mass spec, and this probably is not an accurate description of the fumarate levels in these cells. That's one thing that we've found. All right, so when we look at some of these different metabolites, again, citric acid, if we do a short time course of increasing, short time course of exposure to glucose, you can see citric acid goes up about 30% in the first 10 minutes. So there's a time-dependent increase in citric acid. Glutamic acid, which can feed the TCA cycle, goes down a little bit. Now, one of the things you should notice is the scale here. When you just do relative concentrations of different metabolites, it's a lot of times a little bit misleading on how flux is going. Because glutamic acid in the cell is much higher than the concentrations of citrate, and while citrate goes up 50%, this goes down, but the absolute massive decrease in glutamic acid is actually quite substantial. Malic acid goes up markedly during this time, and I showed you that malic acid actually accumulates with time, and this increase goes from 50, so there's about a 7-fold increase in malic acid. And so if you think about glutamic acid, which can feed the TCA cycle through alpha-ketoglutarate here, so glutamate can be changed to alpha-ketoglutarate. One way to understand this is that massive malate coming in to alpha-ketoglutarate is then just feeding this part of the TCA cycle. Aspartic acid also decreases, and aspartic acid is actually substantial concentrations in the cell, and there's about 100 millimole per milligram protein decrease in the first 5 minutes, and then it sort of rebounds a little bit. So how this all happens is most people just assume that the malate was coming from the TCA cycle, or perhaps through another enzyme called pyruvate carboxylase, which pyruvate comes into the cell, gets carboxylated, and that makes oxaloacetate, which then can make malate. Okay. So that's this pyruvate carboxylase can make oxaloacetate, and then going backwards and filling this site. And this actually has been the main way that people think that the TCA cycle is anaplerotic, i.e. increasing the amount of TCA cycle intermediates in the cell. But when we did fluxomic studies, where we added uniformly labeled C13 glucose to the media and then followed the carbons through, we got a completely different answer, which was kind of exciting because this is the first time that anybody has shown this. Now, if you look at citric acid, what we believe is happening is that what people have believed is that the 2 label here, it's kind of hard to see because I've made this black and white. I should have used the colored slide. But what we look at is the number of carbons contained in the citric acid. And I think Charles Evans on Monday told you about using isotope labeling, right, to be able to see the carbons. Now, citric acid comes from acetyl-CoA, which is derived from glucose. So this glycolysis comes down, you make pyruvate here, and then you make acetyl-CoA, and this is 2-carbon labeled acetyl-CoA that's generated in the mitochondria. And that condenses with oxaloacetate. And so citrate generated from the condensation of 2 labeled acetyl-CoA with oxaloacetate will give you 2 labeled citrate. Okay? And indeed, that's what we see here is that there's an increase in 2 labeled citrate. And there's some intermediates that are higher labeled. It probably represents a turn of the TCA cycle. And there's a slow accumulation of these other metabolites as you turn this TCA cycle and you get more acetyl-CoA 2 carbons being formed. You'll get some 4 labels. You'll get, then when you turn again, you'll get some 3 labels and all that kind of stuff. Okay. Now, interestingly, glutamic acid goes down. There's a little bit of labeling of glutamic acid. This probably represents some exit of the 2-label alpha-Q deglutarate going backwards to glutamic acid. And that goes down, then it rebounds, and then you start filling up with more 2-label. And so there seems to be a rapid consumption of unlabeled glutamate. You can see how this goes down. And then there's a rebound of glutamate, but it's labeled glutamate, and it seems like that probably comes from the alpha-ketoglutarate, because that's the only way that you can practically get 2-labeled glutamate in this sort of time frame. Okay? So what we see is that citrate is getting 2-labeled, glutamic acid is getting 2-labeled, but there is some consumption of glutamic acid. Now, what was really interesting is that the increase in malate, malic acid, was nearly all unlabeled carbons. So early on, you can see that there's about a 3- to 4-fold increase in the amount of malate in the cell, but most of it is unlabeled. And what this suggests to us is that the malate is not coming from pyruvate, which would be 3-labeled coming into the mitochondria, and being carboxylated to make oxaloacetate. If that got turned into malate, you would have a 3-labeled malate. And we see some 3-labeled malate, but very little of it in the early times. In fact, just you can't see it much, but there's only a wisp of that, suggesting that pyruvate carboxylase is not really a very good source of malate to do anaplerosis. When we look at the labeling patterns, it doesn't seem like a lot of it's coming from glutamic acid, although we can't rule this out. There's different ways. We've done some labeling with glutamate, and we see very little labeling of malate in the TCA cycle when we add labeled glutamate to the media. Now, the problem is that the glutamate is actually rapidly turned into glutamine, and there's some confusion about where a lot of this glutamate's going. Some of the glutamate actually may be packaged into granules. There's some evidence that it's taken up into the insulin granules and actually is secreted as maybe part of a neurotransmitter effect. But when we look at the labeling patterns, where we believe that the malate is coming from is the aspartate that is decreased. And what you can see is that there's a rapid decrease in the aspartate levels, similar to glutamic acid. But if you look at the labeling patterns between the two, it looks like the malate and the aspartate are interchanging, that this aspartate's being generated that's coming into the mitochondria, and that entry into the mitochondria is generating malate. Now, why would this be important? I'll come back to the slide. It was supposed to be up and disappear. I guess I got it backwards. What the aspartate malate cycle does is actually takes NADH from the cytoplasm, and by aspartate being changed to oxaloacetate in the cytosol into malate that essentially puts reducing equivalents into malate, which can then go into the mitochondria and then regenerate NADH, which can go into the electron transport chain. And what this does is allow glycolysis to continue going, because if you remember, glycolysis needs to regenerate NAD to continue having glycolysis going. Eric? So, no, not really. One of the things that we did do, and I wasn't going to show it, but there's a phenylacetic acid specifically blocks the malate shuttle here. If we actually block the malate shuttle, which is how malate comes into the mitochondria, we can markedly attenuate the rise in the malate in the cytoplasm. So by using these kind of tools, we can actually show that this is probably why the malate aspartate shuttle is the thing that's driving these reducing equivalents into the mitochondria. And then when as you can see, that a little bit of malate that's going to be labeled will actually then come to equilibrium with aspartate, because this is getting regenerated and coming out. And so by looking at the relative ratios of the label of aspartate and malate, this is how we've concluded that you have a rapid entry of malate in, and that this is probably where most of this malate is coming from. Okay? So without using a label to understand the labeling patterns, we would never have understood this. And this actually, even though People have known that these kind of things happen for the last 25 years. This is the first time we actually demonstrated that pyruvate carboxylase, which can generate malate, is not the way that the anaplerosis goes in the beta cell, at least in this cell line. And we do have some evidence that this happens in intact islets as well. Okay. Now, one of the big problems, so we've done a lot of things, and what I'm showing you here is sort of the final thing that we've seen in the time course of 0, 5, 10-minute stimulation with glucose. What you can see is that the biggest problem with fluxomics, at least in our hands, is really how to represent the massive changes in metabolite levels and the relative labeling of different metabolites with time in order to make sense of all this kind of stuff. And really, this is the problem with fluxomics. Not the problem, it's actually the promise of fluxomics. When people like Santiago and George figure out how to give us the equations to do it, then we just need to figure out how to visualize it. Now, Ala and I have talked about different ways that we might be able to do this by looking at different colors in the program Metscape, and different sizes of balls, and trying to do these kinds of things. But it's just really tough. But we think that this may be a reasonable way to do it. If you actually look at the labeling, you can see the C13 isotopomer levels of 1 carbon, 2 carbon, 3 carbon, 4 carbon, 5 carbon, 6 carbon labels on these different things. And so if we go through this and we go, where's glucose coming in? So most of our fructose bisphosphate, which is derived from glucose, is 6 labeled, right? There's a little bit of 0 labeled, and there is endogenous glucose and other glucose-like molecules that actually are identified there. You can go down, you can see 3PG, 2PG, which are generated and interconvert. You can see is mostly 3 labeled. There's a little bit unlabeled, probably the same unlabeled that's coming from here. And what we've represented here is the increase in mass by the increase in the ball size. The area of the ball actually gives us an idea of how much mass comes in. Then if you come into the acetyl-CoA, what you can see is that there's an increase in 2 label. There's a little bit of unlabeled that's coming in. We're actually a little bit curious about how a lot of this is unlabeled here. And then the citrate is mostly 2 labeled and increases with time. That's the green here. And then you can see all these other things. And so these are the ways that we try and represent at least the flux that's occurring with time. It would be actually much better if we could have a dynamic visualization, but we just don't have that. So these are the kinds of things that we found with this. We've also seen that their citrate comes out, acetyl-CoA, probably some of the acetyl-CoA, the funny labeling is because of the citrate, isocitrate coming out, which regenerates acetyl-CoA, which makes HMG-CoA, which makes farnesyl pyrophosphate, which helps fuse the membranes. And so you can now follow these carbons to these different pathways. Okay, so that's the first thing that you can do, sort of how you can use this to derive novel information by using the right label. Now, you could probe different TCA cycle pathways, and I'll get a reference for everybody. I actually apologize, it's supposed to be on this slide. That there's a great paper if you want to probe different metabolic pathways about the proper labels to use. For instance, if you really wanted to probe the pyruvate carboxylase pathway, using a 2-labeled pyruvate as your carbon source can really give you more insights into how carbons are going through there. Glutamate, you can actually label in different carbons to look at different pathways as well. And so I'll get that reference for everybody so you can actually pull up the paper. It's quite a good paper. It's a little bit dense, but there's a table there that really gives you a great understanding of how to use these different labels. Okay. The second thing that we wanted to do is that there's a phenomena that's out there that fatty acids can markedly increase the amount of insulin glucose excreted TAPA. So if you take palmitate and you add it to a beta cell in culture at low glucose, you get no effect on insulin secretion. Now, when you add glucose, going from 2.8 to 16.7 millimolar, which is the standard concentrations for these cells, you see that you get a marked increase in the amount of insulin secretion. But if you pre-incubate with palmitate at 500 micromolar, which the free concentration is much less, but you can get a doubling, sometimes a tripling of the amount of glucose that you secrete under these conditions. And so we wanted to try and understand what's going on. Why is there a marked increase in insulin secretion? And so this potentiation of fatty acids. And then what I'm going to talk about a little bit later, and we'll come back to, is this whole thing about if you incubate with palmitate for a couple of days in the presence of high glucose especially, you get a marked diminution in the amount of insulin that's secreted. So here's the insulin secretion with glucose. With pre-incubation with palmitate, you can see there's a marked diminution in the amount of insulin that you can secrete. And so this biphasic effect, or this amplification effect, and then sort of a desensitization effect that happens with high glucose is something that we've been trying to understand as well. All right. So again, here's the heat map, and again, how you represent this kind of stuff is sometimes a little bit more difficult. And so what we have here is what happens in a time course, and this is essentially what you saw in the last slide. So it's a different experimental setup, but this is pre-incubated with BSA, and then the time course of increase in these different metabolites or decrease in these different metabolites under the conditions where you had added palmitic acid. And then if you look at the palmate to BSA ratio, so what secretion you got with palmitate and divided by the BSA, you can see the either the amplification or attenuation of different metabolite levels. Okay, and the white star is statistically significant from the baseline values that you get, which is BSA. Now, this is a lot of data, and we're going to come back to some of this. But some of the things to recognize here is that, so here's our time course of insulin secretion without palmitate, and with palmitate, again, we get a doubling of the insulin secretion with a 30-minute pre-incubation. Interesting, palmitoyl-CoA, a pre-incubation with palmitate, not surprisingly, increases the amount of palmitate conjugated to the CoA. When palmitate comes into the cell, it's conjugated to CoA, and that is the active molecule that can be metabolized further. You can see that when you stimulate with palmitate, you get a marked increase in the amount of palmitate. It's much lower, only about 10% level in the absence of pre-incubation with palmitate. But these levels drop about 50% almost immediately upon stimulation with glucose. So there's a rapid consumption of the acetyl-CoA when you add glucose to these cells. You can measure something called palmitoyl-carnitine. And the way you generate carnitines from different fatty acids is that they have to enter the mitochondria to get metabolized. And if you sort of overload the mitochondria, then carnitine will come out. And this is sort of like a pop-off valve. If there's lots of lipids coming into a mitochondria and the mitochondria can't take care of it, what happens is that the CoA that's inside the mitochondria will exchange for carnitine. And it's actually an indirect measure of how much oxidation of fatty acids are occurring in the mitochondria. And what's really interesting is both without palmitate or after pre-incubation, there's a marked fall in the amount of palmitoyl-carnitine being generated in these cells. We believe this represents a marked inhibition of fatty acid oxidation in the presence of glucose coming into the cell. Now, this makes sense in a way. There's glucose, when it comes into the cell, can be turned into malonyl-CoA, which blocks the CPT1, which is the thing that allows, the enzyme that allows fatty acids to enter the mitochondria. So we think that glucose coming in is actually just blocking the amount of palmitate entering the mitochondria. And so the consumption of palmitoyl-CoA is not due to an increase in oxidation under stimulation of glucose. Because that was one of the theories, is that when you put in fatty acids, you just oxidize more, you generate more ATP, and that's part of the increase in the insulin secretions you have. But in fact, what happens, what looks like it happens when you add glucose, you get a decrease in fatty acid oxidation, sort of eliminating the potential for fatty acids to be oxidized and increasing ATP and causing insulin secretion, or augmenting insulin secretion. So we've sort of ruled that part of it out. And then here's the malonyl-CoA. You can see that, interestingly, when you have palmitate in the media, the malonyl-CoA levels actually drop more. And so this is the one part of the phenomenon we still don't understand, why the malonyl-CoA is changing. We have some theories on that. Now, the other thing that we're interested in, and I'll come back to this and why I'm putting this up here, is that glycerol 3-phosphate, which is derived from when you do glycolysis, and you get to that, I'm sure you all remember the dihydroxyacetone phosphate interconverts with glyceraldehyde 3-phosphate, which then can be turned into glycerol 3-phosphate. That's where this glycerol 3-phosphate is being generated from. And so when we incubate with glucose, we get a lot of glycerol 3-phosphate. But again, there's an attenuation of the rise of glycerol 3-phosphate when we put in palmitate, which was, again, it's not quite clear exactly, it wasn't quite clear why this was happening. When we put in labeled glucose, labeled glycerol 3-phosphate, in the absence or presence of palmitate, and we looked at the 3 labeled glycerol phosphates, so when it's coming down the glycolytic pathway and you split the 6 carbons into 2 3 carbons, what happens is the glycerol 3-phosphate being made, it's all being labeled. And what you can see is that everything gets labeled very quickly, but there seems to be a rapid rise in both with palmitate, but the rise is actually blunted. It actually sort of stabilizes and comes to a sort of a steady state. So we're trying to understand how this all happens and how this might be associated with increase in glucose. So I just want to review with you for a second fatty acid metabolism or the use of acyl-CoAs. Glucose gets split again to glycerol 3-phosphate through multiple metabolic steps. And if you have labeled glucose, and there's 6 labels here, you get 3 labels in glycerol 3-phosphate. Acetyl-CoA can be condensed with glycerol 3-phosphate to make lysophosphatic acid. And so what you would have is lysophosphatic acid with it. If it's labeled with glucose, it'll be lysophosphatic acid with M+3. So the mass will be increased by 3 Daltons because of the label that's on there, okay? And then if you do, if you make phosphatidic acid, lysophosphatic acid, I got this label wrong. This is lysophosphatic acid. This is diacylglycerols here. Oh, that's over here. There's a diacylglycerol. This can go to this other place. Diacylglycerols have 2 carbons on it, and this can be turned into some other different things. And these seem to be part of the amplification process. Some of these diacylglycerols have been shown to be important for helping membranes fuse with the insulin granules fuse with the membrane and augment the insulin release. So this very simple slide here, which is with Mahmoud published late last year. What we see is that when we add labeled glucose to the media without phosphatidic acid in here, without palmitate, and we're looking at lysophosphatic acid generation. So this entity here with 1 fatty acid chain on the backbone of the glycerol that has a phosphate, we can identify this in the mass spec. And without fatty acids in here, we get a little bit of phosphatic acid with lysophosphatic acid with an M+3. But when you add palmitate, what you get is a palmitate molecule, lysophosphatic acid with a palmitate molecule on it that has marked increase in the M+3 labeling. So what we believe is happening is when you add palmitic acid to the cell, the glucose is being metabolized, and there's rapid condensation of that phosphatidic, rapid condensation of palmitic acid from the palmitoyl-CoA that makes this lysophosphatic acid. So essentially what's happening is the fatty acid is coming into the cell, glucose is being metabolized, and it's basically grabbing the glycerol 3-phosphate and taking it out of circulation, making this lysophosphatic acid. And so if you go back to this earlier slide, go back the wrong way. Go back to this earlier slide. We believe that this rise in glycerol 3-phosphate without palmitic acid is just accumulation. But this attenuation here in the further rise is because the glycerol 3-phosphate that's being made through the glycolytic pathway is being consumed by the fatty acid and being taken out of circulation. And so you get to a rise, but then everything that's being generated de novo is now being condensed with the acyl-CoA or the palmitoyl-CoA. And that's probably responsible for this marked drop. And so what we're generating is these early metabolites in the lysophosphatic acid. If you look at diacylglycerol, you can see that basically there's no diacylglycerol being made without palmitate. But if you look at the diacylglycerol that has 2 C16 lipids on it, so 32,0, M plus 3, you can see there's a rapid rise in that. So we think that it's all going through that pathway. And if you look at the endogenous fatty acids, you can sort of see the same thing. Well, we can actually see the generation of triglyceride, tripalmityl with M plus 3 label. That comes a little bit later. You can see that this is rapid. This is the second step. It's delayed a little bit. This is the third step that's delayed. And so what you're seeing is sort of a pseudo flux of the palmitic acid into this diacylglycerol and the triglyceride pathway. Now, this is important because then what we do is we generate some of these downstream mediators that are thought to be important for insulin secretion and granule condensation. And so in the end, what we think is happening, and I won't go through the other part of it, but what we think is happening in low glucose or under fasting conditions, which you're all not doing now, presumably you ate a bagel out there, that the long-chain CoAs come in, but with little glucose coming in, you don't have any dihydroxyacetone phosphate being converted into glycerol 3-phosphate. But under the presence of fatty acids, you get a lot of fatty acids coming in. More of the glucose is being shunted over here into and capturing this glycerol phosphate. And there's something called the glycerolipid cycling that helps generate some other intermediates that generate the potentiation of insulin signaling. Now, we think this is really interesting, and it harkens back to this whole thing about the malate aspartate shuttle. So remember, malate aspartate shuttle, what it does is it takes NADH from the cytoplasm and puts it into the mitochondria. The interesting thing is one of the reasons we think that we get a marked amplification, partly a marked amplification, is that when you go from dihydroxyacetone phosphate to glycerol 3-phosphate, that also generates NAD+ in the cytoplasm now. And this may be part of what markedly accelerates glycolysis when you add fatty acids to the cell. So we think that the amplification is both the increase in the glycolytic pathway, because we have shown directly that when you add fatty acids into the cell, you get an increase in oxygen consumption in the beta cell. And this increase in oxygen consumption is not due, as I showed you earlier, due to fatty acids being oxidized, but it's due to the increase in glycolytic flux that's being augmented by the regeneration of NAD to get the glycolytic pathway going faster. So there's two things happening. We think we increase the NADH flux into the mitochondria, increase glucose flux into the mitochondria, increasing oxygen consumption, but also at the same time generating potentiation signals, these diacylglycerols being formed by the fatty acids. So again, you get this convergence effect, and that probably is the basis for this amplification of glucose secretion under conditions of addition of fatty acids. Okay? So again, using these Fluxomics sort of studies, we can actually now follow carbons and going into the right place and seeing which metabolites you generate to help sort of dissect these signaling pathways. All right, so finally, which time do I got left? 10 minutes? 5 minutes, okay. So this will be quick. So the last thing I wanted to show you is decline in insulin secretion that happens. So there's two phases of decline in secretion when you add high amounts of fatty acids or glucose to cells. There's a decrease in insulin secretion, and then long-term, probably in vivo, more apoptosis of beta cells, which leads to diabetes. So if you kill your beta cells, you can't secrete insulin, and then you get type 2 diabetic. And people who are obese and are prone to type 2 diabetes have large amounts of fatty acids and higher glucose floating around their cells, or more glucose hitting the beta cells, and this may be. So we did an experiment where we took and incubated either with high fatty acids or high glucose for 3 or 5 days, and then looked at insulin secretion. You can see that there's a decrease in the amount of insulin secreted after stimulation of glucose with either fatty acids or glucose. And so we wanted to try and understand this. And again, the ability to visualize this, you have to sit down and look at these things, and I won't go through the whole thing. But there's some if you look at the control to free fatty acids, interestingly, there's not a huge amount of difference of different metabolites under these conditions. with fatty acids. You can see that the patterns are pretty much the same across. There's some differences in some of the amino acids, and we'll come back to that. But when you actually incubate with glucose, what you see is that at baseline, there's a marked increase in glycolytic intermediates, and there's a marked increase in the amount of TCA cycle intermediates from the control. But then when you stimulate glucose, you get, in the control conditions, you get an increase in the TCA cycle intermediates, as I've showed you before, a decrease in these lipids and other mediators. But what happens with high glucose, you really get no further increase. And so these black here means that when you add glucose to the cell, you just don't get more of those kinds of things going in, okay? So this is the same thing, and this is supposed to be that. So what we use is Medscape, and this is actually a true story. We didn't understand exactly what's going on. Why both of these conditions, where you get very little effect with fatty acids, but a big effect with glucose pre-incubation. But both of these conditions decrease the amount of insulin that's being secreted once you add glucose back to the cells. So this is one of the earlier things that we did with Ala, with Medscape, and I have to thank her because she did all this work. So what we did is we took the 98 metabolites that we could find, and we put them in a layout, and you see these red dots here are the identified metabolites. We selected a sub-network, and obviously we looked at all of these, but we took, plucking out the TCA cycle here. Somebody asked yesterday, can you put these into where you want them to see a TCA cycle? Where everybody has this visualization, these Behringer-Mannheim maps that TCA cycle has the glycolysis have to go around, and we're all sort of used to that. So we actually just artificially artificially made all this work. And so here's the TCA cycle, and we're interested in a number of different metabolites. And because of our earlier work with alanine and things like that, we wanted to find alanine. Alanine's officially not part of the TCA cycle. So what you can do is, so here, I can't read them from down here. So we expanded the metabolites, and we took oxaloacetate. I think Ala showed you this yesterday. You can find oxaloacetate on here, and you can expand. By clicking on this, you can expand out, and you can get secondary metabolites that are there. And I think aspartate is aspartic acid or aspartate is up there. And then what we can do is animate it and put these side by side. Now, she showed you a little bit differently. So here's the control conditions at baseline. Here's the levels of the metabolites that we see, and these are relative levels across these conditions. And so you can see here that at baseline, there's the TCA cycle intermediates between control and fatty acids are about the same. But at baseline, you can see that the TCA cycle, the main TCA cycle metabolites are much higher after pre-incubation with glucose, even though we took these cells when they were in high glucose and we washed them out and incubated them in low glucose for 2 hours, there still was a maintenance of very high levels of TCA cycle intermediates, even though they were pre-incubated in low glucose. So something has happened such that these things are maintained at high levels. Okay? So what you can do with this is then you can toggle this, as I showed you yesterday, and by changing it, you can see how things change. Now look here and here and here about what happens in these different metabolites as you go back and forth, and you can see here. So you add glucose for I think this case was 25 minutes, and you can see that there's in the control example. This is aspartate. You can see that aspartate levels drop, as I showed you before, after you add glucose. And the aspartate levels don't change in the fatty acid conditions. In fact, when you actually look at it, in the fatty acid conditions, aspartate level is down already. And if you look over here, aspartate levels are really down in the glucose condition. So by toggling back and forth, you can see how things change, and you can see how things are different between these things. Okay? And this actually gave us the first clue that this aspartate may be a really important thing, this decrease. And if you look through, and you can expand these things, and you can now look at aspartate is up here, and glutamate, you can see the different levels. And what's cool about this program is you can actually call up the metabolites when you click on a certain subsection. It'll call up the metabolites, and it'll give you the relative levels. And what you can see now is that compared to 100%, which is the highest level that you're going to see in the cell, Under control conditions, after these 2 time points, citrate goes up. Again, you can see this is the increase in citrate that we saw before. Aspartate levels drop off, and after about 25 minutes, it goes down to about 30% of levels. But if you look at the fatty acids, they're already at 30%, and they really don't decline that much with time. And in fact, in the glucose conditions, the aspartate levels are markedly low. And so we believe that perhaps the consumption of aspartate inside the cell is really part of the process of decreasing the amount of insulin secretion that you get when you reheat it with glucose at this consumption. Now, what's really interesting is that we've tried to add back in a couple of experiments now aspartate to the media to rescue the cells, and we can't do it. And the thing is, we can't get the aspartate into the cells. The levels of aspartate don't rise when you re-incubate with aspartate. And so we think that perhaps the cell is trying to protect itself from all this nutrients coming in by perhaps blocking aspartate taking up into the cell, and this may be part of the reason that you get this dysfunction. So we're trying to figure out exactly what's going on here. So by looking at this and doing the visualization, as Barbara talked to you about yesterday, I'm really kind of a visual guy. And by looking at these things, we were able to really hone in on what may be a very important metabolite for the dysfunction of these beta cells. So now this is not fluxomics. I like to call this pseudo-fluxomics because really it's a time course of change. But we know from the other experiments that we did that the aspartate needs to go into the mitochondria, or at least help shuttle the malate into the mitochondria, and this may be important for this whole effect. And so now that's a graduate student here wants to do a project. I have a great project for you. Anyways, so summary: Fluxomics tells you a lot. Dynamic changes tell you a lot. And so, in my lab, we have a thing, we call this the Heraclitical method of looking at metabolism. Heraclides was a 3rd century BC philosopher who said you cannot understand a river unless it's flowing. Okay? So if a river is still, you can't understand if it's deep, if it's shallow, but if it's flowing, you can really get a whole lot more. And so by doing dynamic experiments and using labels and using time courses, you can derive a lot more information. Now, granted, a lot of conditions, it's really hard to do it, but I really encourage you, if you can think about how to look at changes dynamically, it's going to give you a lot more information. And I'll stop there. And this is all the people who did the work, Matt and Lou especially. Okay, any questions? I know it was a lot. I know intermediary metabolism pretty well. Go ahead. Well, so the level of glucose, it's what's been done in the past in the literature. Okay. We know that at low glucose with these cell lines and in normal beta cells, that you get very little insulin secretion at low glucose. You need to have an increase in flux of glucose coming into the cell. And so now one of the things that I want to and I think I have it in my workshop this afternoon. One of the things that you really, really, really have to be careful of is when you're going to compare different conditions that you do everything the same. So when we add new media back to these to look at the amount of metabolites in cells in culture or cells in suspension, that if you're going to add 16 millimolar or 2.8 millimolar glucose back to the cell, and you're going to look at a time course, you have to do the same thing to the control cells. You cannot just leave those in culture and then look at all these metabolites, because the cells, as they sit in culture, consume all sorts of metabolites. And so you have to really think very carefully about how your control cells that are going to sit at 2.8 millimolar, if you say, well, I'm just going to take the media off this and I'll put in 16 millimolar glucose into those cells, and then we'll look at all these metabolites. If you just use those cells that you didn't do anything with and say, well, they were in 2.8 millimolar anyways, we won't do anything, that's really a bad idea, because what you're doing is consuming the aspartate, you're consuming the glutamate and all that kind of stuff. And so those levels in the cells will have dropped during that next 25 minutes that you have the high glucose in. And so what you want to do is refeed those cells with 2.8 millimolar, and everything else is now fresh in the cell. So think very, very carefully about how you think about the conditions and making sure that you only change one metabolite or a couple metabolites at a time when you actually start doing these kind of experiments. So happy to see you in multiple ways. Anyways, any other questions? Yes, sir.
When you're measuring the movement of the label through the systems, do you actually just look at the MS1 mass change, or do you actually look at change in fragments to actually follow individual atoms?
So we use accurate mass, and so we're just looking at the mass, the shift in the mass of the molecule. So we look for each one of them. Some of them, alpha succinate, there's an overlapping peak at the M+3 and M+4. We really can't see that, but you just look for the isotopomer shift. So the same secretion in the glucose 180.21, and then add M+3 or M+6 if you're looking for the label, and comparing the peak height ratios. And so we put in a lot of these. We put in internal standards so we can actually quantify things. So you look at the M+1, the M+2. For citrate, which is 6 carbons, you look for the M+2. You look for the M+3, and with time, you can see the M+2 is going to go up. The M+3 will start rising a little bit later as you turn the cycle. Yes, ma'am. Yeah, it's a great question. So we did a lot of preliminary experiments, and so when people come use the car, we say, well, you really need to do a time course to see how fast the metabolism is in your cells. If you use primary culture, metabolism tends to be much slower, and so you need to label for a little bit more time. But part of the labeling may be fast and part of it may be slow, and so you really need to do some preliminary experiments to look at the time course. And so we usually ask people to do one or two, a couple of replicates, and we'll do an initial time course just to get an idea of where you want to sample things. Because if you go too long, the labeling patterns just get all mixed up. Now you can actually do, Santiago knows how to do all these magical tricks to be able to look at the steady-state concentrations and derive sort of insights from that. I'm not smart enough to do that. And so we try and do initial uptake studies, and we think in a way you get a lot more. But you do get stuff if you actually come to steady state and get a steady state isotopic distribution. You can make some inferences from that as well. Erica? Yeah. So again, it's actually from preliminary experiments. How well can you extract? When we do this stuff in intact islets, it's much more difficult because the size of the islets are different. The number of beta cells versus alpha cells in intact islets are different, and so. But when you do flux experiments, you get the advantage of doing ratios, and the absolute quantification becomes a little bit less important. And so, but sample size, I think these were N of 5 for each time point. There's 5 replicates in each one. Whether we could have gotten away with less or more, it's really more time and space and statistics more than anything. But but again, I can tell you there was lots and lots of preliminary experiments doing these flux experiments before we really got these nice, pretty pictures. Matt really worked out the conditions under which we can do these, and there was a lot of false starts through all this. But we can help you. We've made all the mistakes, and so again, if you're going to do an experiment and you want to do this kind of stuff, come talk to us, because we can, between Santiago and Michelle, we've done a lot of stuff, and the guys the guys in the lab. We've done. I won't show it today, but we've done a lot of in vivo flux experiments as well, where we can inject glucose or amino acids or different things into animals and actually look at dynamic changes in different tissues and things like that. And those are even more difficult because if you inject and your blood labeling is changing with time, you need to take that into account when you do those kind of things. But we can help you with those as well. Okay.